Hi, I'm Sarah Smay, and this is Chapter 6, Vision. Our learning objective for today is explain how light moves from the retina to and through the brain, including information about the types of information that are processed at each stage in the pathway. Light is the stimulus for visual processing. It is a narrow band on the spectrum of electromagnetic radiation. Electromagnetic radiation with a wavelength between 380 and 760 nanometers is visible to humans. The perceived color of light is determined by three dimensions: hue, brightness, and saturation. Hue is determined by the wavelength of light, brightness is determined by the intensity of light, and saturation is determined by its purity. The eye is the sensory organ that receives light, visual stimulus. It has many functioning parts, all working together to focus the image onto the retina, where it causes changes in the electrical activity that result in messages being sent through the optic nerve to the brain. The sclera is the opaque, tough, white outer coat of the eye that does not permit light to enter. The cornea is the transparent outer layer of the front of the eye that allows light to pass through. The pupil regulates the amount of light that enters the eye. The iris is the pigmented ring of muscles situated behind the cornea. The lens is the transparent series of onion-like layers behind the iris which changes its shape to permit the eye to focus images of near or distant objects on the retina. The retina is neural tissue and photoreceptive cells located on the inner surface of the posterior portion of the eye. Receptor cells, also known as photoreceptors, are in the retina, the rods and cones. Rods and cones detect light. They are named for their shapes. These photoreceptors convert light into neural signals. They both have an inner and outer segment, but they have different functions. The human retina contains approximately 125 million rods and 6.4 million cones. Rods are located in the peripheral retina. They are not found in the fovea. They are sensitive to very low levels of light and provide monochromatic information. They have poor acuity. It would seem that cones are greatly outnumbered by rods. However, cones provide essential visual information about the small details of the world around us. They have very sharp acuity, detect color, light of varying wavelengths, and are found in the fovea. They are sensitive to moderate to high levels of light. The retina is divided into three main layers: the photoreceptive layer, comprising of rods and cones, the bipolar cell layer, and the ganglion cell layer. It is important to note that the photoreceptors are at the back of the retina. Light must pass through the overlaying layers to get to them. The epithelium is located at the back of the retina. It is opaque, so light can't pass through, but there are vital nutrients here. Photoreceptors form synapses with bipolar cells. Bipolar cells form synapses with ganglion cells, whose axons travel through the optic nerves and carry visual information into the brain. Horizontal and amacrine cells transmit information in a direction parallel to the surface of the retina and thus combine messages from adjacent photoreceptors. I'm not going to go into depth on lamellae and photopigments, just make sure you understand the difference between them and the different types of photopigments, but I'm going to quickly go over sensory transduction. Photopigments split into their components, opsin and retinal. This causes a change in the receptor potential. Neurotransmitters are released into the bipolar cells. The cell activity changes, and so does the amount of neurotransmitters released. Neurotransmitters are then released into the ganglion cells, where an action potential is generated and sent down the axons to the optic nerve. These are also pretty straightforward concepts. Make sure you understand the difference between midget bipolar cells and diffuse bipolar cells. In perceiving color, make sure you understand how photopigments are sensitive to a certain type of wavelength and understand the difference between rods and cones in relation to color. And also note that rate of firing determines color perception. After receiving the signal from the eye, the brain must begin to process it. The retinogeniculocortical pathway is the primary pathway that carries information from the eye to the brain. Signals coming from the photoreceptors in the retina are transferred through the optic nerve, decussate at the chiasm, through the optic tract where they are sent to the dorsal lateral geniculate nucleus, LGN, in the thalamus. It is then sent to the striate cortex in the occipital lobe. The LGN is composed of 6 layers. These layers receive input from one eye. The inner 2 layers are known as the magnocellular layer. They are larger cell bodies and transmit information pertaining to form, movement, depth, and differences in brightness to the primary visual cortex. The outer 4 layers are known as the parvocellular layer. These are smaller cell bodies and transmit information pertaining to color and fine details to the primary visual cortex. The conicellular layer is the smallest layer below magno and parvno. It transmits some information from blue cones to the primary visual cortex. From the LGN, the information is then sent to the striate cortex. There are 6 layers within the striate cortex, and they are sensitive to features such as orientation, movement, spatial frequency, and color. Orientation and movement are analyzed by simple, complex, and hypercomplex cells. The complexity of these cells increase as one moves down the layers. This area of the brain also processes spatial frequency, which analyzes different levels of brightness to provide definition of form. Retinal disparity provides depth perception. The striate cortex is a modular organization with 2 blobs. Inside blobs perceive color. Outside blobs perceive everything else: orientation, movement, spatial frequency, and retinal disparity. Some outside blobs are orientation sensitive to detect object edges. Simple cells can detect a line in the center visual field. Complex cells can detect a line in many areas of the visual field, and hypercomplex cells can detect when the end of an object extends outside of the visual field. The visual association cortex, or the extrastriate cortex, V2, surrounds the striate cortex. This is used for further analysis of visual information. Information from V1 is integrated by V2. 20 to 25% of this cortex is devoted to vision. Cells respond to specific features. Information can feed forwards and backwards through this cortex. Regions for visual processing is divided into 2 streams, the ventral and dorsal streams. The ventral stream processes information about form and is referred to as the what stream due to its involvement in object identification. It is composed of magnocellular and parvocellular cells. The end location for this stream is in the inferior temporal cortex. The dorsal stream processes information about spatial location and movement and is known as the how or where stream. It is composed of magnocellular cells. The end location for this stream is in the posterior parietal lobe. This article takes a look at how diabetic retinopathy is the leading cause of acquired blindness. Vision loss, among the most feared complications of diabetes, is primarily caused by the diabetic retinopathy, a disease that manifests in well-recognized characteristic microvascular lesions. The reasons for retinal susceptibility to damage in diabetes are unclear. However, the unique metabolic demands of retinal neurons could account for their vulnerability in diabetes. Photoreceptors are the first neurons in the visual circuit and are also the most energy-demanding cells of the retina. In this study, the researchers review experimental and clinical evidence linking the photoreceptors to the development of diabetic retinopathy. The conclusion: photoreceptors are clearly significant contributors to early retinal disease and diabetes. Collectively, these concepts form the basis for a growing body of investigative efforts aimed at developing novel pharmacological and non-pharmacological tools that target photoreceptor physiology to treat a very common cause of blindness across the world. And here are my references. And that is it, guys. Thanks for listening and good luck on the exam.